Alright, nature of bonds in polymers. The next topic. Polymer which is made up of monomers, right? So in a polymer, it always happens that so many monomers are linked together by specific bonding. Make sure there is always specific bonding for the different monomers. And for bond formation, it occurs by dehydration. It means we have to remove water moiety. Water moiety for the formation of bonds. Now coming to polysaccharides. In polysaccharides, monosaccharides are linked by 1,4 glycosidic bond. Now let's do the formation of this glycosidic bond. Why 1,4? Because that is the position. This bond is formed between the first and the fourth carbon atoms of the successive monomers. I'm taking an example first. Okay, now what is this? It's a glucose molecule. This is the first position, second, third, fourth, fifth, and the CH2OH group, the sixth carbon. Same way, 1, 2, 3, 4, 5, and the sixth carbon. Clear? We are talking about this first and fourth. Here it is HOH, HOH, OH, H, HOH, and CH2OH, H. Same way, HOH, HOH, this is the hydrogen bond and hydroxy bond. Here OH, H, then H, OH. So we have these two, HOH at the first and the fourth position. And we are going to make bond formation here. What will happen now? This is how it appears to be. HOH and HOH at the first and the fourth position. So these two will join together to release a water moiety and get fixed by O. Let's put it back here and let's find out how the structure is going to get complete. Look. This is H, HOH, HOH, OHH, H, and here CH2OH will remain as it is. Then same way, this is how we are going to show that bond is formed. This is known as 1,4 glycosidic bond. Clear? And if we want to show it as a chain reaction, so this is how we take the glycosidic bond formation in a chain. That how this bond is formed between the first and the fourth carbon atoms of the successive monomers in a branched polysaccharide. And here branching is done at the sixth position. So the branching is 1,6 glycosidic linkage. Clear? This is 1,4 glycosidic bond which is taking place in polysaccharides. Now moving to the other one in polypeptides. When we talk about polypeptides, we say successive amino acids are linked by the peptide bond. So which bond comes here? Peptide bond. It's like CH. I'll write it one. That is the first carbon or the first amino acid in the chain. Here NH. It has CO. Then this NHCH, that is the second one. CH2SH. Or maybe we can write CH2OH as the second one. This linkage is the peptide linkage. How is it formed? We have first carbon as written in this form, first amino acid. Okay. Now this is the second amino acid. CH2OH, here N, H and H. Alright. This side again C double bond O, OH. So we have the first and the second amino acid. But we are going to take out water moiety from here. When water is eliminated, the entire structure takes a different name and acquires a bond. This is the new structure which is formed. Got it? This is known as peptide linkage. I have written CO, NH and then continuing with the second amino acid and first amino acid. Alright, this is the peptide linkage. Here successive amino acids are linked by a peptide bond which is formed between the carboxy group of one amino acid and the amino group of next amino acid with the elimination of water moiety. It is represented as CONH bond. So moving to the third one, that is the phosphodiester bond in nucleic acids. When we say nucleic acid, here the successive nucleotides, these are linked by phosphodiester bond. And here the linkage is a little different, as it is the linkage of 3' carbon of sugar of one nucleotide with 5' carbon of the sugar of next nucleotide. Okay, and the bond formed between the phosphate and the hydroxyl group of sugar is an ester bond. Where the bond is formed, it is between the phosphate and hydroxyl group of sugar. So we call it phosphodiester bond because it is an ester bond and it is at both sides, so we call it phosphodiester bond. And this bond is formed like this. This is first, second and the third position. Okay, the third position. This is fourth and this is the fifth one. So third is first of all making a bond. This is phosphodiester bond. Between the successive nucleotides. The structure initially is like this. This is the fifth carbon. This is the third carbon. 1, 2, 3, 4 and 5. Here we have this hydroxy group. And the successive element CH2OH of the next nucleotide lies here. From here, this water is eliminated and the phosphate group, it takes its place to complete the structure. This structure is formed on both the ends or both the sides of the nucleotides. This is the first carbon. Here is the first carbon. First carbons are supposed to have nucleic acids, that purines and pyrimidines. It may be adenine, it may be guanine, cytosine, uridine, any of the pyrimidine or purine here, that is always attached at the first position. But third prime or the fifth one, carbon, they are supposed to make a bond which is known as phosphodiester bond. And this phosphodiester bond provides a stable structural stability to this nucleotide structure. And the nucleic acid is formed. Next we move to the dynamic state of body constituents, that is the concept of metabolism. What is metabolism? Metabolism means a sum total of anabolism and catabolism. Clear? Now, basically biomolecules means that they are constantly being changed into some other biomolecule and also being made from some other. It means they do not remain static in their own manner. They keep on changing. And these metabolic reactions do not occur in isolation, but are always linked to some other reaction. And such linked reactions are known as metabolic pathways. And flow of metabolites in this is called metabolic basis of living, because if we consume food, let's take a simple example, we have consumed carbohydrates. Do they remain carbohydrates as it is in the body? No, they are broken down into the sucrose one, or maybe we'll say disaccharides, monosaccharides, right? The change into one or the other metabolite in this pathway is known as metabolic reaction. And these are metabolic pathways. There is the flow of metabolites in this reaction, right? And when we talk about we say all the chemical reactions, they follow anabolism and catabolism. Anabolism, which are formed from simple ones, like amino acids are forming proteins. Catabolism, where a complex is broken down into simple one. These proteins are broken down by enzymes into simpler amino acids. Peptidase will work, protease will work. So energy for these reactions is liberated from a compound called ATP, adenosine triphosphate, also known as the energy currency of the cell. Now when we talk about a living state, we say these biomolecules in any living cell are in a metabolic flux. That means the state is non-equilibrium, which is able to perform work. As soon as we attain equilibrium, we cannot work. We always keep on trying, the bodies always keep on trying to achieve equilibrium, and as the moment we reach equilibrium, we attain equilibrium, we cannot perform work. So basically it means we will die, because all the reactions will be at the steady state. They will become constant. It is a constant effort to prevent reaching equilibrium, which is achieved by energy input. Since metabolism provides the mechanism for the production of energy, the living state and metabolism are synonyms. Without metabolism, there cannot be living state. Metabolism means anabolism and catabolism. Food is taken and broken down, or the smaller particles, they make up a bigger particle. This is in relation with the living state. If body is not showing anabolism and catabolism or metabolism, that means body is not working anymore, right? So we are always trying to achieve it. We always try to achieve equilibrium by energy input or output. But once we will achieve equilibrium, we will not be able to work. Body will not be able to work any further. Clear?